In the last session, I mentioned that there are countless microorganisms around us, with bacteria and fungi being representative examples. These microorganisms are surrounded by a cell wall. Today, let's explore the structure and role of the cell walls of bacteria and fungi, as well as their differences. First, the cell wall of microorganisms consists of a cell membrane and peptidoglycan in the case of bacteria, and a cell membrane and glucan in the case of fungi. The movement of substances due to osmotic pressure or the movement of substances and stimuli through membrane proteins occurs through the cell wall. Additionally, a proton motive force can arise based on the cell wall. So, shall we learn about the structure of bacterial cell walls and their differences? Bacteria are classified into gram-positive and gram-negative based on differences in their cell wall structure. For example, Staphylococcus is a gram-positive bacterium, while Escherichia coli is a gram-negative bacterium. Structurally, gram-positive bacteria are characterized by having a thick peptidoglycan layer instead of an outer membrane, while gram-negative bacteria have a thin peptidoglycan layer along with an outer membrane and LPS layer. These structural differences can be clearly observed through electron microscopy. Moreover, there is a gram staining method that utilizes these structural differences to distinguish between gram-positive and gram-negative bacteria. The gram staining method involves staining with crystal violet, fixing with iodine, decolorizing with ethanol, and counterstaining with safranin. The key principle during the staining process is that in gram-positive bacteria, which have a thick peptidoglycan layer, crystal violet remains, while in gram-negative bacteria, the thin peptidoglycan layer allows the crystal violet to wash away, resulting in staining with safranin. When performing gram staining, gram-positive bacteria appear purple, while gram-negative bacteria appear red or pink. In particular, the outer membrane of gram-negative bacteria contains lipopolysaccharides LPS, which are known to play roles such as cell protection and immune system activation. However, recent studies have revealed that LPS from some gram-negative bacteria can cause respiratory inflammatory diseases and sepsis. Fungi, such as molds and yeasts, have a different cell wall composition compared to bacteria. Instead of a peptidoglycan layer, they are composed of glucan, chitin, and mannoproteins, which is a key difference. So far, we have confirmed the differences in the cell wall composition of bacteria and fungi. Thank you. This has been Microbalance.